Today we have three speakers. Professor Sidney Brenner will start this afternoon with his lecture, followed by Professors Urs Satmari and Gerd Müller. I think when we talk about Professor Brenner, we should not say too much since everybody here in Singapore knows Dr. Brenner, who is Singapore's Nobel laureate. He's world famous for his discoveries, primarily his work related to Caenorhabditis elegans, for which he got the Nobel Prize. And of course, during his long professional history, he has been working with giants like Francis Crick or Jim Watson, and he contributed to the birth of molecular biology with numerous seminal and pioneering discoveries, including the description and the deciphering of the codon and the triplets. All these works together resulted in his Nobel Prize in Physiology or Medicine. And I purposefully put something here on this slide. You may see that he even discovered the name of the most famous Swedish orchestra of the time of his Nobel Prize, ABBA, ABBA. So Sidney, thank you also for naming ABBA in your landmark paper describing the codon. Of course, Dr. Brenner has contributed with several seminal discoveries and books to our present understanding of biology in general and molecular biology in particular. Thank you, and the word is yours, Sidney.
Thank you very much. Can you hear me? Thank you. So thank you very much for that introduction. And what I'm going to do it's not going to be a long lecture, so you don't have to fall asleep during it. What I'm going to do is to try to describe the approach that I've taken to the subject, and to try and reduce it to a much more simple understanding than you can get if you study the papers that have been published. There must be well over 1,000 papers on the subject. And of course, what we are interested in is, of course, the genetical theory of selection. So this has quite a long history when it was realized that somehow these genes that existed inside all animals and plants were going to be the ones that were important in determining and in the changes of the organisms from the very beginnings to the very end. And we have this information. We don't have many old genomes from long ago. There are very few fossil genomes that you can study in detail. Svante Pääbo has done excellent work on the bits of genome that have survived from hominid, from humans that have predeceased our time. And the other material that we have are, of course, the fossils. And that is indeed another subject. But we will always go back to that because many of these remains can be dated because of the geological conditions that existed at the time. So what we would like to know is, if we start with the most primitive whole cells. When did they actually start? And I'll start with prokaryotes. If we look at prokaryotes, we can find that there are, in general, two kinds of these, distinguished by certain physiological properties. They are, one of them is called the archaeobacteria, and the other are the normal bacteria. And if we look at these, just taking a look at how many genes does it take to make a bacterium, the basic number appears to be of the order of about 1,800. 1,800 genes makes up, for example, a primitive a primitive bacterium that can photosynthesize and create everything. That occurs in the oceans. Probably the biggest photosynthetic force in the world today, converting carbon dioxide into oxygen. Another bacterium can be found which can exist on many small molecules and create most of these from elementary chemical processes. They have about 1,200 genes, which includes everything that is required for the cell membrane, for making all the proteins that are used in these very complex chemical transformations. And then if we look at all the other bacteria that exist, we find that there are bacteria that will range up to 6,000 genes. That is, they're usually about double the normal thing. And of course, it's of great interest to look at these. And I'm doing this in order to show you what it is that you need in order to evolve. You need new genes. You need to make genes that are different, that can do other kinds of chemistry. And as you will discover during the course of this, the way this is done is either to steal them from other bacteria or to duplicate your present gene content and allow deviations to occur with novel consequences. So I just want you to notice one very important thing which explains a lot about a bacterium like E. coli. Now, why do we concentrate on E. coli? E. coli has found a way to live in two worlds. It can live inside animals, as it does in our intestine, and it can also live in the free, open space. And how does it accomplish this? Because the most expensive part of the machinery of all cells is the creation of all the material, the ribosomes, and all the other enzymes that are required to copy nucleic acids and to translate the copies into protein sequences. This is a very elaborate structure, and 25% of E. coli's weight is simply ribosomes, which is a very elaborate machine that allows you to place amino acid after amino acid in the correct order. All right. So, as you may have realized, it takes a certain amount of time for E. coli to make another E. coli. It has to copy everything, and it then has to separate the copies into 2 cells. So that every time E. coli can get something for free, like an amino acid or a protein or a nucleotide, the system has evolved so that automatically, the biosynthesis is switched off. So that it is a characteristic of all amino acid pathways is that they are switched off by an allosteric mechanism, as it is now called, by the product of the reaction pathway. That immediately stops the consumption of raw material. Secondly, there is yet another step by a special protein that acts to turn off the production of messenger RNA from that gene. And that is why there are repressors. That is why they are stopped. As soon as the organism is repressed, and one can make measurements of this for each element of the pathway, as soon as that is repressed, more is gained in the distribution of all the energy and material requirements, so the cell can grow faster. And E. coli has then a huge amount of apparatus simply to regulate the biosynthesis. Now, of course, that evolved quite separately from all of this, but it so allowed these extra genes, E. coli, to have an ecological, if you like, balance between living for free inside an organism and getting lots of things for nothing, and living outside where life is pretty tough and you have to make everything. So the evolution of regulation, of doing this rather than that, both of pathways and of protein synthesis, is very old and comes soon after the creation of the first free living organism. So I've given a little bit of that background because that's how we have to look at everything that precedes. Everything there is done in order that the organism can fit its environment better, because that is the principle of natural selection. And you will find huge examples of this where organisms actually employ other organisms to help them survive. And so the control mechanisms that have arisen have always been in response to this business of a fitter organism for that environment. and sometimes employs using other organisms, sometimes definitely employs using new genes that appear on the site. Well, as you know, this is the basis of what we are going to discuss and try to do, and that's been the basis of all of the discussion of the genetical theories of natural selection and evolution. And a tremendous amount has been written on this subject, some of which involves quite abstruse statistical arguments, but we're going to ignore all of that for the moment, and just show that a lot of it is unnecessary. So once we started to sequence proteins, once we started to do all of that, it soon became clear that proteins didn't have a fixed structure, that if you looked at the early days of cytochrome C, hemoglobin, myoglobin, all of those gave rise to showing that there were amino acids that came in and were a measure of the change, if you like. And one of the first theories that had been done to try to go a little bit further than the Darwinian principle was produced by a man called Kimura. Kimura produced a theory of what he called neutral selection theory. He noticed that most of the changes occurring in proteins, most of the differences in amino acids, most of the changes, therefore, in the genome were of this neutral kind. And they couldn't possibly explain what he was looking for, which is in the evolution of the whole apparatus like this. And so he produced a theory which said most of the changes were neutral. That is, had no contribution to fitness. So it was just accidental, so to speak, that they were put in there. And actually, he found that much later, and I just tell you this because you can forget about it, that the only way they could explain evolution through a neutral theory is to assume that the neutral changes were actually deleterious, partially, likely. There's a very elaborate concept of this. So I have asked another question, which is much in the same line, but tries to remove the confusion produced by the advocacy of Kimura-type theories. We can ask ourselves this question: what changes in the genes would leave the organism exactly the same as it was before? So this is, if you like, absolutely neutral mutations. The reason for this comes out of the genetic code. I'm just going to tell you part of this story so that you can begin to see how this is done. So if we look at the codons, that is the triplets that stand for the various amino acids, we see that there are several kinds of these. Right? And we see that, in particular, they differ largely in the third position of the codon. So both, to give you one example, both TTC and TTT, differing in the third position, a C for a T for a C, code for phenylalanine. Now, I should make it clear that there are many cases where there's only one transfer RNA that does both. And it's done by what Francis called the theory of wobble. Now, it's actually in detail is accomplished by slightly more than this, because oftentimes the actual detecting base is modified. For example, guanine is changed into inosine, and inosine can pair with both cytidine and also with adenine. So you can then interpret A and G as exactly the same base, because inosine can pair with adenine by a kind of slightly different Watson-Crick base pair. So that we can forget about that and just say that if I changed the G in the C in your anticodon in one of these groups of codons, if I changed it to a T, nothing would happen. You make it be exactly the same, at least for the protein thing. And that, I think, is the key to how we're going to do this analysis. So we can go through all the proteins that are made by everything and classify the codons into a number of sets. I'll just deal with the codons that do two things, and two things only. Some of them can read all four, but I'll leave those on the side. The two codons that can be read are all the ones that in which you read A and G. So these are called 2-star, two cases. They're called 2-star AR. R means A or G. So it's just simply says what fraction of these R codons are A. The rest of them are G. And that change, which is a G to an A or an A to a G, is without effect on the proteins of that animal, whether it's human or a bacterium. Similarly, we can do exactly the same with T and C for another set of codons. All right. Now the mutation between these codons are called transitions. All right. And the interesting thing is that for these two sets, the transitions are exactly the same, must be the same. They must follow the same rules, simply because G is the base pair of C and A is the base pair of T. So we have two things which we can work out which must be the same, and anything different about them can be checked out, as you will see. So this, leave the others for the moment because it's a little bit more complicated. So what we can do is go through all the genomes that have been sequenced, well over 100, I mean, not only of bacteria, but of humans, chickens, fish, anything that has been sequenced, crocodiles, and you can convert them all into the pattern of these two classes of codons. All right. That is a transformation that's easy to do. All right. Now what we can do is we can ask, if I take the codons of each class from each gene, what is their average? Because we'll find that the codons from some genes are very high in C or G. And from other genes, they have absolutely or mostly A. So we can describe a scale, the average scale with which the codons from a given gene are on the scale. And it's done on a scale of 100, okay? And they are binned into groups of 4 and labeled. So we have 25 of these, with each containing 4 codons, which on average differ by 1 extra A as we go down the list, or 1 extra T. All right. And now what we do is just take all the genes and do this with them. So you sequence the genes from a bacterium, you can transform them into this. Okay. So it's just a set of figures, all right, that you get for each gene. Maybe a few thousand genes, there may be 20,000 genes. All right, you just do it that way. Now, having done that, you ask yourself, what is the distribution of this amongst the genes? That's just a matter of doing a little bit of sorting. And what you will find is that when I take the whole genome and plot it, I find a lot of genes of this class and a lot of this class and so on. And I can make then a map of the organism which shows me the distribution of these genes on a scale of 1 to 100 in 25 groups of 4. Okay, now the interesting thing is that since the change in a gene and in a genome is under one rule of transition, right, I can now say that the more mutations I have, the more that gene has been evolving, or the more the gene has evolved, the faster the gene has evolved. All right? So now we can ask ourselves, if I do this and I say, I'll take, in bacteria are all different because this ratio, this transition rate, varies according to various rules within a gene. So we have bacteria, which, like the streptomycetes, which are very high GC. And we have bacteria at the other end, which are very high AT. And that has remained fixed. It's remained fixed because it's fixed since the last time it got fixed. I have no idea how long the streptomyces have been like this, but I can tell you they've been a long time, because the rules of mutation fix the limit of this distribution. As you realize, if there is reverse mutation, as soon as you have too many of the things, you get beyond the equilibrium, you will go backwards. So even when the genome doesn't change, the distribution for streptomyces is the same, whatever streptomycete we do, okay? Then even if that doesn't change, there still is mutation, because the changes are going round and round all the time. If it goes beyond the equilibrium, then they just go backwards. The reverse mutation then controls everything. All right? Now, so what we do is we can take organisms like bacteria, and we can say, well, since the rules I've given you are exactly like this, what happens if I plot these distributions against each other? Okay? Because one is very high GC, the other is very high AT. Okay, and it tells you that you should have reached a steady state. All right? You should reach a state which you can draw a map of. That map is a set of circles, okay, a contour map of the genome. It's perfectly round, it's perfectly symmetrical, or should be, according to this theory. And it is then, you can just divide by how many genes are in any particular class. So this is a map of a genome with changes in it. All right, now, why should you be able to get that map? Because the theory of random processes predicts that the mutations should occur as a Poisson thing, and when continued, they will eventually form a normal distribution. That is inevitable, all right? And the normal distribution is symmetrical, all right? It rises. So at the midpoint, where 50% of the data is, on the left-hand side, A or T is increasing, whereas on the other thing, on the other side, it's G or C is increasing. It's going backwards, all right? And this should then be the theory. So you can take bacteria, you can take lots of them, make these maps, and to one's slight horror, one discovers that there are deviations from it. All right, now, that is, some of these maps are elliptical. They ellipses, they tilted, as though one's changing faster than the other, but to the same extent. All right? So they are deformed. Now, we know that very well from other activities. It's called, and I'll just do it, it's called biased gene conversion. And we know how that works, actually. Now, it so happens that if we went to yeast, we'd find a lot of that. All the yeast things are tilted because of biased gene conversion. But we can explain that easily in yeast because yeast has hotspots for recombination. At recombination, the protein is, and the recombination, the thing is severed, it's cut, it's chewed back, and it's repaired. So you always are copying your strand at that point. You're copying your strand off the old one. You're putting in more G all the time, feeding in more G or more C at the time, depending on this. All right? So that the biased gene conversion doesn't mean I'm biased, because I need to be biased to explain why there's so much high GC genes in my genome. It just is because they are the genes through which recombination takes place. All right. But you can say, how can you use that for bacteria? They are haploid. They only contain one genome. So, but it turns out, and this is a discovery I made only a few weeks ago, that when a bacterium replicates, the two new double helices are wound around each other, okay? And they must be broken and separated. Furthermore, that when the two new genomes, which are circles, are exposed to the other chromosomes, are segregated into different progeny, there is exactly a similar breakpoint and exchange of things. So you are diploid, if you like, at the end of your replication. And that explains perfectly. So we can explain all bacteria very simply by this mode of what is the mutation rate, which is fixed according to the metabolism, and what is the segregation. So bacteria are perfect objects and will go on changing, and all are at steady state. Now, I can actually do the same with the human genome. I can ask myself, if I take all the genes, just those parts from your genome, and look at them in this way, how can I describe the resulting things? Well, what I discover is it hasn't got very far. It's nowhere near equilibrium. It'll take a long time to come to equilibrium. If I do the same for the mouse, I find actually that mycoclosa, that is the long sausage, is now becoming more and more a broader sausage. And the only organism so far that we have found of the higher organisms is the hagfish. The hagfish is very close to equilibrium. So the hagfish has either very old, or is both old and fast. Okay. It turns out that if I take plants and I do the same thing, cabbage, you just name it, all the leafy plants, they're all at equilibrium and all can be described in this way. And they form beautiful circles that we can do it. But plants have only been in existence they only started round about 200 million years ago, perhaps even later, plants as plants growing on the earth. So how did they get to equilibrium? So that is one of the first clues that you have to get rid of when you start to think about this. The reason is that this counts generations. It counts every time you make new gametes and bring them together. And that happens once a year because of the seeds being produced and because the lifespan of a plant is essentially 1 year. So when we start to count now in generations, we can now see why things are so different. A mouse lives about 3 years, 2 to 3 years. A human is more than 10 times that, just 20 times that. So the mouse is evolving 20 times faster than the human being. So it looks pretty old, okay, because it's come a much longer way. But that's just a confusion of the way we are counting this. And so, in the end, what I hope to do is to be able to construct a complete map of every organism that has been sequenced. I can already do this, and in order to say what has exactly happened to it during the course of its existence and evolution. Now, once we've got that, then we can start to ask real questions about evolution. We've got the neutrality out of the way, so you can forget about Kimura and all those people. It's just interesting historically. This is a much better way of looking at the evolution. And of course, you all know the answer. Evolution has got to do with the creation of complexity. And the simplicity of these, it's got to do with the creation of developmental pathways. You can't just build an organism about one kind of cell. And the big difference between us and our cousins, the chimpanzees, because our genomes are 95% identical to the chimpanzee genome in this part in the proteins, with few exceptions. So, but it turns out the chimpanzees had a genome that was like ours 25 million years ago when they appeared first. Okay, we just went on a different path during that time, where the chimpanzees started to decay by mutation along this another gradient. So we should then be able to put all these organisms on a map. And the most important thing in evolution is the creation of new genes. So much so that we believed that when we did the genome of the octopus, we would find it had a duplication. But it didn't. The octopus doesn't. But spiders have duplicated, and so have crabs. Crabs and spiders are related, as you know. They're all in the same class. So this duplication is quite important. And as you know, in the humans, the vertebrates, you not only have a duplication, you have a second duplication. And amongst fish, we have even a third duplication to give us new copies of genes. And it is these, then, that we have to use to understand what has happened in evolution. And I think that doing this in detail is very important, because then we are constructing events that have very significant points of change. One mutation can do it all for certain things. And that means that we need to have a skeleton on which this can be based, which I say we have, and then we can get on with the rest of the job. Right, now, I just finished with one statement. As you can see, I'm not saying that this is why, this is my theory of the propulsion. I'm just trying to say, let's try and see what is the general process of genome changes. Okay. But there are lots of other changes that, of course, we try to find an explanation for. And I think you should take this view, which is the one I'm proposing, that we're going to publish our papers in a new journal called the JCC. The JCC stands for the Journal of the Crumbling Cookie. Because for many things, all you can say, that's the way the cookie crumbled. You can't say why, it just happened that way. And our job is to separate those issues. As to, people have asked, why are there only two sexes? Why aren't there 3 or 4? I've had friends who have experimented, not practically, with several new sexes, but that's the way the cookie crumbled, that's the way the machinery works. You just got to say, that's it, finished, no more discussion. And I think we need to be able to see what is pure chance, because that's the game. The game we're playing, there's nobody sitting anywhere saying, hey, you better do this, or you better do that. It's pure chance. And pure chance then explains this great progress in evolution. I hope that I will finish this work in the next, by the end of this coming year, when then I might even stop working on evolution. Thank you.
Thank you so much, Sydney. And now questions from the audience. Please don't be shy. A unique opportunity. Yes.
So it's an absolute honor to be in the same room as you. My question, I hope it doesn't turn out to be a JCC. My question to you, sir, is that what is the purpose of all the non-coding genome that exists in our bodies? And if there's no purpose, why has nature not eliminated it already? Thank you.
I'll tell you what the purpose is. The purpose is to confuse you. It's obviously fulfilled the purpose. There's no purpose at all. If it doesn't do anything to the organism, it'll still be there. It might disappear by chance. It might not. That's the way the cookie crumbled. So that you have to accept that we don't have a pure tax on garbage. Our genomes seem to be okay when they are very big. Just look at the genomes of salamanders. The chromosomes, they have something like 20 times the amount of DNA of frogs. They have huge chromosomes. They have huge cells. You can almost see them with the naked eye. So it just works. And the purpose is so to give you an opportunity to ask me the question.
Thank you.
Questions? Yes, there is a question.
Hi, Sydney. Can you tell me where you stand on this archaea to eukaryotic transition? Are archaea the mothers or the sisters of eukaryotes?
I can't hear. What does this say?
The archaea to eukaryotic transition. Where do you stand on this?
Archaeas to eukaryotic transition.
What?
So are archaea the mothers of eukaryotes, or are they the sisters?
archaea.
Yes.
Yes. What is the question?
In your opinion, are the archaea the mothers of eukaryotes, or are they the sisters of eukaryotes?
I don't understand.
Are they mothers or sisters of eukaryotes?
Mothers or sisters?
Yes. So where do you have the branch in evolution?
That's a later stage. They all got together and decided they would make a new organism called a eukaryote. So parts of it came from the archaea and parts came from the ordinary bacteria. That's all. In fact, in many cases, they're both parts there. If you want to see it, look up the biosynthesis of carbohydrates of arginine and the other amino, the amino acids, because both systems are there. Okay, one happens, and often does happen, to reside in mitochondria, but sometimes they leave the mitochondria and get back into the genome. So you just have to, it's an agglomerate.
Further questions? If not, let's thank you, Sydney, for this spectacular afternoon.
Just